When identifying plant sampling sites, it is a good idea to go back to existing soil sampling sites to plant sample in season. This helps to complete the full nutritional picture of the paddock and helps to monitor and ensure that the fertiliser recommendations and applications generated from soil sampling are on track and correct. Both existing and new plant sampling sites can be identified and selected using Decipher Ag. These sites can then be sent to the Decipher Ag mobile app for collection. Alternatively, plant sampling sites can be created and selected in the paddock using the Decipher Ag mobile app. For each sample collected, you will need to complete the sample information on the Decipher Ag mobile app. This can also be done manually if you're not using the app. The sample information needs to be filled out accurately and comprehensively. The information contained will determine the accuracy of both the interpretation and the new Logic fertiliser recommendation. You'll have the option to reuse the information from the previous site or enter new information when using the Decipher Ag mobile app. The information that CSBP requires is the crop type. The new Logic model is calibrated differently for the different crops and pastures grown in Western Australia. The date of sowing and date of emergence allows us to calculate the age of the plant, which helps in the analysis of the sample results. Without this information, it is very difficult to accurately interpret the results. Crop yield target gives an indication of the production target of the paddock and ensures that the most suitable recommendation is given after the results are analysed. Plant component refers to the part of the plant that has been sampled. Some crops and pastures require different parts of the plant to be sampled to give the most accurate results. In cereals and canola, you can sample the whole tops, which is the whole plant minus the roots, youngest mature leaves on canola, or youngest emerged blades in cereals. Sampling only the youngest emerged leaves or the youngest emerged blades will give an accurate indication of sulphur and microelements only. When sampling lupins, it is a good idea to sample the main stems at flowering, and pastures require the leaves and petioles of subclover plants and the leaves and stems of ryegrass plants. Entering the crop growth stage helps us to further determine the nutritional status of the plant and the plant size in relation to the age of the plant. The next piece of information required, and the most important piece of information when sampling whole tops, is the number of plants. The number of plants will determine the individual plant size and the dilution or amount of nutrients in that plant. Without an accurate plant number, the interpretation of results may be flawed. The number of plants that you sample will be determined by the size of the plants. It is good practice to sample no less than 20 plants when sampling cereals and no less than 10 plants when sampling canola. The smaller the plants are, the more plants will be required to make a representative sample. It's also a good idea to select a number of plants to be sampled at each point in the paddock and sample the same number at every other point in the paddock. This will reduce confusion and ensure that the information is correctly entered once the samples reach the lab. Comparative growth will be determined by whether you are doing a whole paddock for monitoring or whether you're diagnosing poor growth areas compared to good growth. Entering the soil type information also helps to give some context to the whole paddock when generating a recommendation from the results. Other information about recent fertiliser or chemical applications is also important. When plant sampling, you need to ensure that you have the correct equipment, including your smartphone, with the latest version of the Decipher Ag mobile app. If the job has been planned on Decipher Ag, ensure that you have sent it to Decipher Ag Mobile whilst in mobile range. You'll need a pen for labelling your plant sample bags. You'll need plant sampling bags for your plant samples to go in. It's good practice to label the plant sampling bag with the trading name, the site name and the paddock name. You'll need rubber disposable gloves. It's important to wear gloves at all times when plant sampling. We're testing for very small amounts of micronutrients, and contamination can be transferred from our hands to the plant samples from things such as sunscreen, fences, metal tools, ag chemicals and the like. You'll need a pair of sharp, clean secateurs for cleanly cutting off the roots of the plants from the whole tops. It's also important to make sure that the crop you're sampling has not been recently sprayed with any chemicals or liquid fertilisers, both from a contamination and a personal safety point of view. If any chemicals have been sprayed, it is a good idea to wait at least 1 day before sampling the crop. This information needs to be recorded in the sample information. So to navigate to the sample site, you need to fill out the information on the sample bag, then use the Decipher Ag mobile to complete the sample information and scan the sample bag barcode. To begin sampling, you need to randomly select a plant and pull the whole plant out of the ground. You need to ensure that you pull out the complete plant, including the roots, taking care not to tear off any leaves or stems in the process. You need the whole plant in the sample, otherwise the size of the plant will not be accurate and the accuracy of the results will be compromised. Repeat this process, randomly selecting plants and walking in a zigzag fashion until you've pulled up enough plants. Carefully separate the plants to count them and ensure that you are counting individual plants only and are not separating plants at the base and counting tillers. When starting out, it is good practice to take your time at this point to ensure that your counting is accurate. Take care to only randomly select plants when sampling, and when counting, do not discard any plants, as you need to ensure that you have a sample that is representative of the paddock. Only selecting the biggest or smallest plants will skew the results and can affect the recommendation. Another method of sampling is to count the plants as you pull them out, taking care to only remove individual plants from the ground. As long as you're taking care to get an accurate count of the plant numbers, it doesn't matter which way you sample the plants. Do whatever you find most comfortable. Once you've counted the plants, you will need to cut the roots off neatly with your secateurs. In cereal plants, cut the roots off where the plant stem turns white just above the crown roots. Cut only the roots off. Cutting too high will reduce the plant weights and potentially affect the results. Let the roots fall to the ground and ensure that there is no dirt remaining on the bottom of the stems. Dirt can contaminate the sample and affect the accuracy of the results. Sample photos can also be taken at this point if desired. Once you've removed the roots, put the plants into the sample bag and fold the top of the bag over to make sure that the plants don't fall out before placing the bag in the plant sampling envelope. If the plants don't fit in the bag, it is fine to either bend or even cut the plants to make them fit. Just make sure that all the plant material ends up in the bag. If the crop is wet, take the time to gently shake off as much moisture as you can before putting plants into the bag. Please ensure that there is no plant material hanging out of the bags before you post them. You can then select Finish on the app and navigate to the next site and repeat the process. Random sampling is imperative. Be representative and select at random from within the paddock. Your plant numbers must be accurate. Take your time to ensure that individual plants are separated and counted. Ensure that your sample information is correct. Gloves need to be worn at all times. There can also be no roots or soil present on the plant material. And ask yourself, has the crop been sprayed recently? And is the crop wet? Plant samples should be posted the same day of sampling or the next day at the very latest. Talk to your local CSBP area manager to organise your sampling requirements this season.